This video covers section 5.1 of chapter 5. Here are the learning goals for chapter 5. We want to learn about the methods of binding ligands and proteins. We'll look at quantitative and graphical modeling of protein-ligand interactions. We'll discuss the interaction of globins with oxygen and non-oxygen ligands. We'll discuss the physiological regulation of oxygen binding. Then we'll move on to looking at the mechanism and control of antibody-antigen interactions and muscle contractions. There are two types of interactions that proteins have with molecules. One type involves an alteration of the configuration or composition of the molecule that the protein is interacting with. And that is descriptive of an enzyme, which is what we'll talk about in chapter 6. Now in this chapter, we're going to discuss the other type of interaction where neither the configuration or the composition of the molecule is altered. Interactions of the second type are important: oxygen transport, immune function, and muscle contraction. There are some key principles to start off with. The first is that the function of many proteins involves the reversible binding of other molecules. And when a molecule is bound reversibly by a protein, it's called a ligand. And the important word there is reversibly. A ligand binds at a site of a protein called the binding site. That's pretty straightforward. And finally, the interaction is specific, meaning a protein can discriminate amongst thousands of molecules and selectively bind one or just a few. Now, any given protein may have separate binding sites for several different ligands. And the binding of a protein and a ligand is often coupled to a conformational change in the protein. Interactions between ligands and proteins may be regulated, usually through specific interactions with one or more ligands. In section 5.1, we will focus on two classic proteins, myoglobin and hemoglobin, which are both part of the globin family of proteins. And we will use these to illustrate how protein-ligand binding occurs. These two proteins were the first two proteins for which three-dimensional structures were determined. They both bind oxygen, which is important since oxygen is poorly soluble in water and cannot diffuse through tissues. We can think of the binding of a protein and a ligand as a reversible transient process of chemical equilibrium. And it can be represented by this equation: A plus B yields AB. The molecule that binds to the protein is called the ligand. It's typically a small molecule. And the place where the protein binds it is called the binding site. Now the binding is going to occur via non-covalent interactions, the same ones that we see that dictate protein structure. Now let's consider a process in which a ligand, L, binds reversibly to a site in a protein that we'll call P. So here's just a cartoon. So here the ligand fits into the binding site of the protein. Now we can talk about the forward reaction and the reverse reaction. So the forward reaction we can give a rate constant to, and we would call that the association rate constant. So that's going to tell us the rate of the ligand binding to the protein. Now the reverse, we would give the rate constant, the dissociation rate constant, Kd, and that's going to give the rate of the protein-ligand complex dissociating and forming just the protein and the ligand by itself. Now after some time, the process will reach equilibrium where the forward and the reverse reaction rates are equal. And we can also write an equilibrium constant expression for this. So again, we can just think of this like a normal chemical reaction where the equilibrium constant expression for any typical chemical reaction is the products over the reactants. So here we can write a mathematical expression for the equilibrium constant, and notice I'm using a capital K. That's distinguishing that K from the little k's I use to indicate the rates of the forward and reverse reactions. So the Ka would be equal to the concentration of the protein-ligand complex divided by the concentrations of the reactants, or the protein by itself times the concentration of the ligand by itself. Now we can then relate that to the equilibrium dissociation constant, which would be looking at the reverse reaction. And then if we include that in this expression, then Ka is equal to 1 over Kd. So Ka is the equilibrium association constant, and Kd is the equilibrium dissociation constant. So what this means is that if you have a high Ka value, then your protein and ligand exist mostly in their complex bound form. But if you had a high Kd value, then the dissociated forms would be favored. Now in practice, we often determine the fraction of binding sites, which we sometimes denote as theta or as Y. And we can define those as the amount of ligand bound to the protein divided by the total of ligand bound to the protein plus unbound protein. Now remember, we have this Ka expression. If we substitute the concentration of PL with Ka times L times P, we can eliminate PL, and then we rearrange to give the result in terms of the equilibrium association constant, such that the fraction of occupied binding sites is equal to the concentration of the free ligand divided by the concentration of the free ligand again plus 1 over Ka. And then remember, 1 over Ka is just Kd, so we can just rearrange the equation again and put it in terms of the more commonly used equilibrium dissociation constant. So as a result, we get to this final equation where the fraction of occupied binding sites, theta, is equal to the concentration of the ligand divided by the concentration of the ligand plus Kd. What this means is that the fraction of bound sites depends upon the free ligand concentration and Kd. Now let's think about this equation. If Kd was equal to L, then the fraction of bound sites would be exactly 1/2, because the equation would be L divided by 2L, which is 1/2. So Kd can be determined by looking at a curve, a graph of the fraction of bound sites versus the concentration of ligand, and determining the concentration when the fraction of bound sites is exactly 0.5. So here, if we look at this curve, the x-axis has the concentration of the ligand, the y-axis has the fraction of bound sites. At 0.5, when half of the protein is bound by ligand, we can then drop this down to the x-axis and see what the concentration is there, and that's going to be equal to the Kd. So the concentration of the ligand at which half of the available ligand binding sites are occupied is equivalent to the Kd. So let's try an example. Suppose I gave you this graph and it has two different curves for two proteins A and B that bind to the same ligand L, and they produce the binding curve shown. So can you figure out what the dissociation constant is for each protein? And then can you figure out which protein has a greater affinity for the ligand? So let's look at the curve for A to begin with, and we're going to look at the y equals 0.5 point on the curve, which is right here. And then I'm going to see what that correlates with on the x-axis. So that would be 2 micromolar. So the Kd for protein A is 2 micromolar. I'm going to do the same thing for B. So at B, that curve crosses the 0.5 line right here. And then when I drop down to see what that correlates to on the x-axis, that's about 6 micromolar. So the Kd for protein B is 6 micromolar. So what does that mean regarding which protein has the greater affinity? So you have to remember, if it has a higher Kd, then that means it prefers to dissociate more. And affinity is looking at how much the protein wants to associate with the ligand. So in this case, A has a half saturation point at a lower concentration of L, which means it has a lower Kd and therefore a higher affinity for the ligand. So another way to think of it is that it didn't take as much of the ligand to be present for protein A to have half of its binding sites filled. That means it really wants to bind to the ligand more so than protein B. With protein B, it took a lot more ligand to be present before half of its sites were occupied by ligand. Now let's look at an example of oxygen binding to the protein myoglobin. So this is a cartoon of myoglobin. It contains 8 helices, which are labeled A through H. And then here in the center is the heme, which I will get into more in a minute. And that is where the oxygen is going to be bound. So that's where the oxygen, which is acting as the ligand, is going to be binding to myoglobin. Now you may also notice all of these bends and turns, and they are labeled according to the different helices that they connect. So this one connects A and B, so it's called bend AB. This one connects C and D, so it's called turn CD, etc. Now we can make a graph just like we saw in the previous example, plotting the fraction of bound sites versus the concentration of the ligand. Now the ligand in this example is a gas, it's an oxygen molecule. So instead of using concentration in terms of moles per liter of solution, we'll use the partial pressure of oxygen. But we're going to do the same analysis. So when we look at the 0.5 mark on the y-axis, when half of all of myoglobin's binding sites are bound by oxygen, we can see that that corresponds to a pressure of oxygen of 0.26 kilopascals. Kilopascal is just a unit of pressure. What this means is that myoglobin binds very tightly to oxygen. Now we can also relate the equilibrium association or dissociation constants with Gibbs free energy using these equations. So if I gave you the KD, you could figure out the Gibbs free energy for that particular protein ligand binding event. Now back to some generalities about proteins binding to ligands. There is a lock and key model that was proposed by Emil Fischer back in the late 1800s, and he said that a protein and its ligand bind to each other like a lock and a key fit in together. And only certain ligands bind, and that high specificity can be explained by the complementarity of the binding site and the ligand. Now this model assumed that the surfaces, the ligand and the protein, were pre-formed and ready to interact with each other. They were in the proper conformation at the outset. However, in 1958, Daniel Koshland proposed that actually the lock and key may not be exactly right. Conformational changes may occur upon ligand binding, and that adaptation is called induced fit. And that induced fit allows for a tighter binding of the ligand and for a high affinity for different ligands. And in this situation, both the ligand and the protein can change their conformation when they come together to get to a better fit. Now back to globins, which are oxygen binding proteins. How do proteins bind oxygen? Well, the side chains lack affinity for oxygen, so that is not how they bind them. So instead, prosthetic groups such as heme are used, which contain an iron atom that will coordinate to an oxygen molecule. And both myoglobin and hemoglobin bind oxygen via a protein-bound heme. Heme is a structure that includes a protoporphyrin ring, shown in A, bound to an iron atom, and the porphyrin ring bound to the iron is shown in B. So a protoporphyrin has these pyrrole rings, these 5-membered nitrogen-containing rings, covalently bonded to each other via a methane group. And there can be various substitutions on these rings. Now the nitrogen atoms of each of the 4 rings coordinate to an iron atom, which is located in the center. Iron has 6 coordination spots available to it, so 4 of those coordination spots are being taken up by the 4 nitrogen atoms of the protoporphyrin. That leaves 2 other coordination spots available. And those coordination spots will be perpendicular to the plane of the protoporphyrin ring system. And C is just a space-filling model representation of B. So B and C are just different ways of representing the heme. So now if we just zoom in on that iron atom, we have it connected to the plane of the porphyrin ring system that's occupying 4 of its 6 available coordination spots. What happens with the other 2? Well, there is a proximal histidine residue in myoglobin in helix F that's going to coordinate to iron in one of the 2 spots. That final spot is where oxygen is going to bind. So notice in this figure where the heme is located, it's essentially in the pocket made up largely of the E and the F helices. And incidentally, about 70% of the amino acid residues in myoglobin are found in the helices. Carbon monoxide has a similar size and shape to oxygen, and it can also fit into that same binding site. In fact, it binds heme over 20,000 times better than oxygen because the carbon in carbon monoxide has a filled lone electron pair that can be donated to vacant d orbitals on the iron 2+ atom. The protein pocket decreases the affinity for CO, but it still binds about 250 times better than oxygen. This is why CO, carbon monoxide, is really toxic, because it will compete with oxygen. And if it does that, it will block the function of myoglobin, hemoglobin, and mitochondrial cytochromes. This figure shows how oxygen binds to iron and how carbon monoxide binds to iron in free heme. So this would be heme that's not bound to protein. Now, CO would like to bind in this perpendicular fashion. However, when it binds to the heme, when it's part of the myoglobin, it's forced to adopt a slight angle, which actually weakens the binding of carbon monoxide to heme in myoglobin. So this explains why, if you were just looking at free heme, carbon monoxide would have a binding affinity 20,000 times more than oxygen. But when we're talking about heme that's part of myoglobin, the affinity for CO is 250 times stronger than oxygen, which is significantly less than that 20,000 times. And this is important for our physiology since we do not want to bind carbon monoxide. And this picture is just a zoom in on the heme, showing how the oxygen will be bound to the iron, and then the histidine is also going to be coordinated to that iron atom. And then there's a partial negative charge on the oxygen molecule that actually will then form a hydrogen bond with this distal histidine labeled as E7, which means it's the 7th residue in helix E. And this hydrogen bond stabilizes the complex, and that interaction is not present when carbon monoxide is bound, which leads to the drop in affinity of free heme for carbon monoxide versus the bound heme. Now you may have noticed that the heme was deeply buried inside the protein with few pathways for oxygen to enter, but luckily the protein is not rigid, and this movie shows that with rapid flexing movements of the amino acid side chains, transient cavities for the oxygen molecule are made available, and that's how it enters the protein and gets bound to the iron atom. Now this is the graph that I showed you a few slides ago, comparing the fraction of bound sites of myoglobin to the partial pressure of oxygen. And I told you that the KD was 0.26 kilopascals. Now, if the pressure in the lungs is about 13 kilopascals, so we're like way over here, that means that a lot of oxygen is going to be bound. Pretty much all of the sites will have oxygen bound to them. Now, the pressure in the tissues is about 4 kilopascals. So if we look at the curve here, That also is at a very high point on the curve that shows that almost all of the sites available for oxygen to bind will be occupied by oxygen. But then that also brings up a problem because we want to bind oxygen, but eventually we want to release it. We do need to get the oxygen to the tissues. So myoglobin doesn't seem like it's going to be able to do that well. It's going to always just be hanging on to its oxygen and never releasing it. So that's where hemoglobin comes in. So myoglobin is great at storing oxygen. Hemoglobin is our key to transporting oxygen. So nearly all of the oxygen carried by the blood in animals is transported by hemoglobin. And in arterial blood passing from the lungs through the heart, hemoglobin is about 96% saturated with oxygen, but in the venous blood returning to the heart, it's only about 64% saturated. So what happened to the oxygen there? Well, it was released to the tissues, which is very critical to maintaining life. Here is the structure of hemoglobin. It is a tetramer, meaning it has 4 subunits, and the 4 subunits are really a pair of 2 identical subunits, 2 alpha chains and 2 beta chains. The alpha chains each have 141 amino acid residues. The beta chains each have 146 amino acid residues. There are strong subunit interactions in hemoglobin. So the alpha subunits are called alpha 1 and alpha 2. The beta are called beta 1 and beta 2. And the strongest subunit interactions, which are highlighted, occur actually between unlike subunits, between the alpha and the beta subunits. If we look at the structures of the myoglobin and the beta subunit of hemoglobin, we can see that there is striking similarity. And that's despite the fact that the amino acid sequences are identical at only 27 positions. And the hemoglobin subunits also have several helices, which are also labeled with letters like the helices in myoglobin. So I mentioned before that myoglobin is very good at storing oxygen, but not so great at releasing oxygen. So for a protein to bind oxygen in certain places and release it in others, it must have an affinity for oxygen that varies. And this is exactly what we see with hemoglobin. So this would be the type of sigmoid curve here that we see in a protein that has varying affinity. So it could transfer from the low affinity state to the high affinity state depending upon the pressure of oxygen. So as the pressure increases, we go from a low affinity state to a high affinity state. And notice where this would line up with the tissues versus the lungs. So in the tissues, we'd be in that low affinity state where you're releasing oxygen to the tissues. But then when we get to the pressure in the lungs, we're in that high affinity state where we want to bind oxygen from the lungs. So hemoglobin is a lot more sensitive to the small differences in oxygen concentration between the tissues and the lungs, and that's why it can bind in the lungs and release in the tissues. So how can hemoglobin switch between these low affinity and high affinity states? Well, it does this by alternating between 2 conformations that are known as the T state and the R state. The T state is the low affinity conformation. So when it's in this state, it has a low affinity for oxygen. The R state is the high affinity state. So when it's in the R state, it really wants to bind oxygen. So the transition from the T state to the R state shifts the subunit pairs substantially, affecting certain ion pairs. Most notably, histidine HC3 is involved in an ion pair in the T state, but when it's in the R state, it is no longer in an ion pair. Another dramatic result of the T to R transition is a narrowing of this pocket. So notice how this becomes more narrow in the R state. This movie shows the hemoglobin moving from the T state to the R state and back. How can affinity to oxygen change? Well, first of all, it must be a protein with multiple binding sites, and those binding sites must be able to interact with each other through a phenomenon called cooperativity. With positive cooperativity, the first binding event increases affinity at the remaining sites. You can also have negative cooperativity where the first binding actually reduces affinity at the remaining sites. So here is a picture of what that would look like. So first you have no ligand. Much of the molecule is either flexible or somewhat unstable. Then you have ligand binding to one of the subunits. This binding stabilizes a high affinity conformation, and more of the structure is stable and none is unstable. Then the rest of the polypeptide takes up a higher affinity conformation, and this is stabilized in the other subunit through protein-protein interactions. And then a second ligand molecule binds, and that binding occurs with a higher affinity than the binding of the first one. And this is an example of a form of induced fit. We can quantitatively describe cooperative proteins using something called the Hill equation. So if we think about a protein with multiple ligand binding sites, we would describe that with this equation. P plus NL, where N is the number of ligands that can bind to the protein, gives you PL sub N. And then we take the concentration of the products over the reactants to get to this Ka expression. Now when we then rewrite that in terms of theta, we get to this description. Now we can take the log of both sides, which gives us the Hill equation, which is in the form of an equation for a line, y equals mx plus b. So here this would be y, this would be b, this would be x, and then the n value would be the slope, which is called the Hill coefficient. So if n is equal to 1, there is no cooperativity. If it's greater than 1, there's positive cooperativity, and if it's less than 1, there's negative cooperativity. So this shows you the Hill plot of myoglobin and hemoglobin. So for myoglobin across the range of pressures of oxygen, there's no cooperativity, but for hemoglobin, in the high affinity state, there is a cooperativity where NH equals 3. Cooperativity is a special case of allosteric regulation. An allosteric protein is one in which the binding of a ligand to one site affects the binding at a different site, and it can be positive or negative. If it's homotropic, then the normal ligand of the protein is the allosteric regulator. If it's heterotropic, then a different ligand affects the binding of the normal ligand. So cooperativity equals positive homotropic regulation. Now let's examine more the R and T states of hemoglobin. So T stands for 10th state. In this state there are more interactions and it's more stable, and there's a lower affinity for oxygen. R is the relaxed state where there are fewer interactions, it's more flexible, and there's a higher affinity for oxygen. When oxygen binds, it triggers a conformational change from the T state to the R state. And this involves breaking ion pairs between the alpha 1 and beta 2 interface. So this is a close-up view of a portion of hemoglobin in the T state without any oxygen bound. So the interactions between the ion pairs histidine HC3 and aspartate FG1 of the beta subunit are shown, and then also between lysine C5 of the alpha subunit and the same histidine HC3 of the beta subunit are shown with the dashed lines. Now pH also has an effect on the ability of oxygen to bind to hemoglobin. So here we have 3 different curves at 3 different pHs. So are you noticing here, does acidity increase or decrease the Kd? So again, to find the Kd for each curve, we look at the 0.5 value on each curve. So at a pH 7.6, that Kd to me looks like it's, I don't know, maybe 2.7. Then at the curve for pH 7.4, that Kd looks like it's closer to 4. Let's call it 3.8. And then if we look at the curve at 7.2, that Kd looks like it's above 4, maybe 4.3 or so. So as the pH decreases, the KD is increasing. What that means is that as the pH decreases, as acidity increases, the affinity for oxygen is lessening. Actively metabolizing tissues generate H+, which lowers the pH of the blood, which then would decrease the affinity of hemoglobin for oxygen. So in those cases, that's where hemoglobin would be releasing oxygen. The hemoglobin affinity for oxygen depends on the pH because H+ will bind to it and stabilize the T state, that's the low affinity state. And it does so by protonating a particular histidine residue known as histidine 146, which then forms a salt bridge and leads to the release of oxygen in the tissues. And the pH difference between the lungs and the metabolic tissues increases the efficiency of the transport of oxygen. So that is just further emphasizing the ability of hemoglobin to release oxygen in the tissues and to bind it in the lungs. And the effect of H+ and CO2 on the binding and release of oxygen by hemoglobin is called the Bohr effect, which is named after Christian Bohr, a Danish physiologist who first discovered it in 1904. Now, CO2 is also produced by metabolism in the tissues and must be exported. And in fact, 15 to 20% of CO2 is exported in the form of a carbamate on the amino terminal residues of each of the polypeptide subunits in hemoglobin. And the formation of a carbamate yields a proton that then further contributes to the Bohr effect. And the carbamate additionally can form salt bridges, further stabilizing the T state. So what I'm saying here is that CO2 and H+ both will favor the T state and decrease the affinity of hemoglobin for oxygen. Now, there is one other factor to consider in how hemoglobin binds oxygen, and that's this molecule 2,3-bisphosphoglycerate shown here. So this is not a normal ligand, so this would be a heterotropic regulator, and it's present at millimolar concentrations in red blood cells, and it's produced from an intermediate in glycolysis. It's a small negatively charged molecule that binds to the positively charged central cavity of hemoglobin and stabilizes the T state. So this is a picture of it binding to the central cavity in the T state. It will not bind in the R state. Now, this molecule allows for oxygen release in the tissues and adaptations to changes in altitude. Now, in normal human blood, the concentration of BPG is about 5 millimolar, but that's at sea level. At high altitudes, it increases to be about 8 millimolar. The reason it increases at high altitudes is because at high altitudes, oxygen delivery declines by about 1/4 to 30% of the maximum. Therefore, an increase in BPG concentration decreases the affinity of hemoglobin for oxygen, allowing it to be delivered to the tissues at the percentage that is normally delivered at sea level. The final topic is sickle cell anemia, which is due to a mutation in hemoglobin. In sickle cell, a glutamate residue is switched to a valine residue in the beta chain of hemoglobin. And the new valine side chain can bind to a different hemoglobin molecule to form a strand similar to the amyloid proteins that we discussed in Chapter 4. This sickles the red blood cells. Untreated homozygous individuals generally die in childhood, but heterozygous individuals exhibit a resistance to malaria and experience much less severe symptoms. And it is thought that the evolutionary adaptation for this is that it does give an advantage to people for resisting malaria, and if you're just heterozygous, then you can still live with the disease as long as you avoid chronic stress or excessive exercise. And this movie shows how that one exchange of the glutamate for the valine results in these strands that can form. So there's the valine, and now it's going to interact with a second subunit. So now that's formed an octamer, so it's two of these hemoglobin tetramers, and this can continue to bind to other tetramers via additional valine hydrophobic pocket interactions, which then results in strands that deform the red blood cell, giving it a sickle-like appearance. In summary, the affinity of a protein for a ligand is typically expressed in terms of the equilibrium dissociation constant. The lower the Kd, the greater the affinity of the protein for its ligand. The Kd for a protein can be determined by graphing the fraction of bound sites indicated by capital Y or a theta versus the concentration of the free ligand. Both hemoglobin and myoglobin bind oxygen, but only hemoglobin binds oxygen in a cooperative fashion, where it switches between a T state and an R state. The T state is the low affinity state, the R state is the high affinity state. It can also bind carbon dioxide and H plus at different sites, and it binds those with an inverse affinity compared to oxygen. And when CO2 and H plus bind hemoglobin, that stabilizes the T state and encourages release of oxygen. So that's going to happen in the tissues. And then there's also the BPG, which can regulate the affinity of hemoglobin for oxygen as well.